So thank you very much for the introduction, and also thanks the organizers for the invitation. I will now provide you some insights in the current status of microRNA therapeutics in this translational session. I think, of course, it's clear that non-coding RNAs became increasingly interesting because we know that the majority of the human genome transcribes non-coding sequences, and microRNAs have been the earlier ones studied, while meanwhile also long non-coding RNAs and circular RNAs gained increasing interest. And you see this also from this publication record that microRNAs already were studied extensively in comparison to the other newer species. microRNAs, just for the very few of you who may not know, to recapitulate that microRNAs are processed by the enzyme complexes and finally incorporated in the RISC complex to induce translational repression or mRNA degradation. And I just can stress this over and over, that the beautiful thing with microRNAs, but also the difficult things for clinical development, is that in contrast to siRNAs, which target ideally one mRNA, microRNAs target hundreds of mRNAs. Therefore, they control changes in gene expression networks rather than single genes. This may be an advantage in chronic diseases where we have multiple pathways being dysregulated, but maybe also a disadvantage because you may have natural off-targets or targets you don't want to target with microRNA therapeutics. There had been many studies on microRNAs in cardiovascular disease, in the heart, including microRNAs targeting cell death and metabolism of cardiomyocytes, fibrosis, vascular aneurysm and atherosclerotic disease progress, regeneration and reprogramming, meaning the reprogramming from fibroblasts to cardiomyocytes, and finally also androgenic microRNAs. Several ones now, meanwhile, had been translated in a way that they had been either used in more, let's say, pig models or more translation-relevant models, and also to humans. This microRNA 132, is controlling autophagy. Again, we have learned from Mauro that this may be an interesting target in cardiomyocytes, and I will show you later on one of a data slide of the human study using anti-miRs against this microRNA. Also, microRNA 21 now, meanwhile, with Rabea and Stefan Engelhardt, has been published in JHK, and also Christian, our chairperson, is being the lead author here in these pig models, showing that these antifibrotic effects can be recapitulated also in large animal models. And my favorite model still is the paper in Nature, where it has been demonstrated that microRNA 199 indeed can promote cardiomyocyte proliferation, which is one of the first evidence that this can be achieved in a large animal model. We have been focusing on angiogenesis and vessel growth, and what I plan to do today is to drive with you through the journey of the discovery microRNA-92A into translation and give you an update on where we are currently standing. The interest in microRNA-92A goes back a while, so this also shows you that translational work really takes a long time. So we were starting with microRNA research in 2007 when we identified microRNAs in endothelial cells which may control angiogenesis. And already in this screen very early on we found the microRNA 1792A cluster being highly expressed and a potential relevant in endothelial cells. And by doing subsequent studies, we found out that microRNA-92A controls angiogenesis and functional recovery of ischemic tissues in mice. This is just a summary of these data. So we screen for microRNAs that are regulated in diseased heart and also on microRNAs that controls endothelial cell functions. And as said, this microRNA-92A was shown to block angiogenic sprouting and vessel formation most efficiently in spheroid models. These are in vitro models to study endothelial cell growth, which were planted by adding precursor of this microRNA in matricial plaque models, and also in zebrafish we could illustrate an impaired function of the vasculature in these settings. Of course, we didn't want to harm the vasculature, we wanted to promote vascular growth, and we used several strategies, including so-called antagomers, which are single-stranded antisense molecules linked to cholesterol, or RNA-based anti-miRs later on. Both work nicely in the heart of mice, but the dosage necessary to achieve inhibition is less in the RNA-based 92A. You see here very efficient inhibition already with 0.5 milligram per kilogram in the heart of the mice after one single injection, while with antagomers, although well tolerated and also very efficient and functional relevant data were gained it took more 8 milligram per kilogram to achieve the same effects. So suggesting that maybe this is more easy to translate because off-target effects or toxicity of RNAs are of course dose dependent, and the lower you can get, the better. We use these antagomers and later also antimers in mice models first. So this is a chronic ligation of the myocard at the left artery. And you see here that if you block microRNA 92A by antagomer 92 injection, this was done 3 hours after ligation of the coronary artery, you see a preservation of tissue. So it's not only that angiogenesis kicks in, but also we could demonstrate that we could protect cardiomyocyte survival, and this consequently resulted in an increase in contractility as shown here with the antagomer 92A to the 2 control groups. Also, these pro-angiogenic effects were further on recapitulated in hind limb ischemia models, showing also that the perfusion of ischemic legs were increased. So this is a laser Doppler flow measurement, and you see here the ischemic legs in the PBS control group, in the antagomer 92A control group, and the significant increase. As mentioned, microRNA targets are difficult to define. There are several validated targets in respect of the vasculature. Certainly 31, which is the deacetylase, also important for endothelial cell survival and function. Integrin alpha-5 and the transcription factor KLF2, all leading to an upregulation also of endothelial nitric oxide synthase, which, as you know, is a very important enzyme in endothelial cells to maintain functional activity. But I would like to highlight there are many more targets involved in endothelial cell function, and I will come to this point later on. With these data, we went on and teamed up with Christian Kuppert and Rabia Hinkel, and you have seen some of the data which Rabia later on did with the diabetes model. This first study was to assess whether inhibition of microRNA may be protective in an ischemia reperfusion model. You know the model from Rabia likely, and we did apply the LNA after reopening the artery. We used the LNA-based molecule, as said, because it was more efficient with a very low dose. So it was 5 milligram per kilogram per heart rate, so it's quite low concentration. We tested also different application forms, intravenous, retrograde, and intercoronary, and we showed that intercoronary clearly increases the concentration of the anti-miR in the cardiac tissue, suggesting that there's a 6.4-fold enrichment, which may be good if you want to directly apply an anti-miR in the future, which has maybe also other toxic effects or maybe has systemic effects you don't want to. So this may be a way to be used also in other microRNA therapeutics or RNA therapeutic studies. We also showed that particular intercoronary application was well suited to protect the cardiomyocytes and reduce infarct size. Retrograde infusion also worked, but a little bit less efficient, and the intravenous infusion in this dose, which was, as I said, very low, was not very efficient, but also because we have a very strong difference in the concentration in the target tissue. We also validated all of these studies in genetic models. Of course, the RNA-based antimers and antagomers might have also other effects. These are RNAs, so they could bind to something. But also, as you can see here in the global knockout mice, lack of expression, the war motion score, which was used here to assess cardiac function, was maintained. While it was worsened in the wild-type mice, it was preserved in the knockout mice, demonstrating that this is really a proof of concept that genetic and pharmacological inhibition works fine. Of course, as said, it's very important to understand your target and very difficult with the set of microRNAs. It's particularly difficult for microRNA 92A because, as shown here, it's expressed in all different cells of the heart. So in cardiomyocytes, immune cells, endothelial cells, and fibroblasts express this microRNA. So in theory, these antimers could affect targets, different targets also in the different cell types. And together, this could influence cardiac function. To get some hints, we felt that it's a need to really isolate the different cells, because if you take the whole heart tissue, you may miss the specific targets. So if you isolated cardiomyocytes, endothelial cells, fibroblasts, and immune cells in mice hearts are being either treated with PBS after acute myocardial infarction or LNA 92A at day 3 after AMI, RNA sequencing shows that the anti-miR is very efficient in all 4 cell populations, and also that the enrichment works. So these are endothelial cells, these are endothelial cell markers, these are fibroblast markers, these are the fibroblasts, these are the immune cells having hematopoietic markers, and the cardiomyocytes were a bit contaminated. You see here we have some endothelial contamination, but still a strong enrichment for cardiomyocyte genes. Then we looked into the targets and also in the gene expression patterns in the different cell types. And it was very surprising to us to see that these are now the significantly upregulated genes, so the potentially de-repressed genes in the different four cell types. And only 14 genes were regulated in all cell types, while many more than 70 and 80 genes were differentially expressed in endothelial cells and cardiomyocytes. We felt maybe these are indirect genes, but even if you select then the predicted targets, these are predictions based on the sequence, you still see only very few genes being commonly regulated and the majority being specifically regulated in different cell types. So at least on mRNA levels, we have a quite striking cell type specific effects of the LNAs. How can this be? So of course we were wondering, we were expecting more common targets, but of course you may have expression heterogeneity. If you have a very lowly expressed mRNA, of course an anti-miR can't change expression. There may be alternative 3' UTRs where the microRNA binding site may of course be effective or not effective. There may be a competition with RNA binding proteins which are also cell type specific. There may be a competition with other RNAs which compete for the microRNA, or of course there may be also indirect regulatory effects such as a transcription factor which is only relevant in one cell and not in another. We're still working on this bioinformatically to sort out how much the different models contribute. It looks at the time being that 3' UTRs and these RNA binding sites have the strongest impact, and I will show you one example of a 3' UTR, which is differently expressed between endothelial cells and cardiomyocytes, and we think that this may explain the cell type specificity. This was that we found in endothelial cells quite specific responses with respect to autophagy genes. So only in endothelial cells, but not in cardiomyocytes, ATG4A and ATG9B, these are autophagy genes, are upregulated. You can here see the direct comparison of endothelial cells and cardiomyocytes, so no effect here, but an induction in endothelial cells. And we could validate this in luciferase assays that is a direct target in endothelial cells by blocking miR92A using upregulation and the overexpression induces the downregulation. And it turned out that endothelial cells have a longer 3' UTR where the miR92A can bind, while cardiomyocytes had a shorter 3' UTR. And we subsequently, of course, found this interesting because autophagy wasn't described as a potential biological effect of anti-miRs, but it showed us that endothelial cell autophagy can be increased, and this may be protective as well. This is just a validation of the studies in vitro. So LNA92A induced autophagy. You see here LC3B1 processing, and also in vivo in the tissue slices we could see a colocalization index in the LNA92A treated sections. Of course, with these data, you may ask what to treat. So where do you go with these data? Of course, acute myocardial infarction is a nice model, and we have the PIC data, but of course, acute myocardial infarction mortality is very low. We have 2% of bad outcomes after acute myocardial infarction if the patient comes to the clinic. And also with the cell therapy studies, we have seen that this is very difficult to improve. You need thousands of patients to see any statistical effect. So we and others, of course, looked also in other disease models where the vasculature is important. And beyond the AMI studies, there are now data, these had been old data in atherosclerosis, where inhibition was protective. And we have more data in diabetes, wound healing models, that microRNA inhibition is also protective. And there's also interesting study in other tissues, for example, in glomerulonephritis, which might be an additional interesting model to block miR92A, which would have a beneficial effect here. And of course, Rabea has shown you now the nice PIC data with diabetic cardiomyopathy, or let's say also cardiac injury and this disease. This could be an interesting target. as well. Finally, because we are looking into heart failure, as you know, heart failure with preserved ejection fraction is one of the unmet medical needs in the cardiac field. Very high mortality, as high as HFrEF, you see here, and also increasing incidence. So we wonder whether the effects on the vasculature may be protective if we block miR-92A in a HFpEF model. This is a mouse model established by Joe Hill. So we treated in these mouse models after two weeks after start of the diet with LNAME and high-fat diet. We started with LNA92A versus control treatment, and nicely could show that diastolic function certainly was improved. So there was a preservation of this diastolic function in these mice in comparison to the LNA controls. You see here the statistical significance at 10 weeks, suggesting that this might be an additional, very interesting unmet medical need which we could use the anti-miRs to target. Finally, of course, you may need to know certainly whether the anti-miR can be safely applied and is efficient in humans. Actually, anti-miRs, there's not so much known about the efficiency also in humans. And with the help of the DZHK Translational Research Program, we were able to really perform an intensive study over years to do tox studies, GLP tox studies, and finally ending up in a first-in-human study, which I shortly will present you now. First of all, we checked also how to optimize the RNA-based anti-miRs, and it was interesting to note we couldn't play with the sequence because this is of course given, but we could play with the RNA-DNA positioning, and it turned out that the RNA positioning has effects on the efficiency. You see here A, B, C, these are 3 different positioned RNAs, and you see that there is a more efficient and less efficient RNA, and also ejection fraction was better in the A compound, and we selected this then for further subsequent in vivo studies in humans. And this is now the first-in-man study, which we did together with Xavier, where we injected healthy volunteers. So this was the prerequisite of the Paul-Ehrlich Institute and the regulatory agencies, that we should use dose-dependent escalation in the healthy volunteers first before applying this in patients. We did blood samplings pre-dose and then during the first hours and weeks. And these are the results now. So we used whole blood, we isolated RNA. You see here the miR-92A 3P in the whole blood, and you see a nice dose dependency. Please don't forget, this is a logarithmic scale, so this is all very efficient, inhibited with the higher doses, and higher doses means quite low doses. So above 1 milligram per kilogram, we had almost 100% inhibition of the miR in the first hours in the whole blood. Of course, this doesn't tell you so much because if you only have the substance, and we wonder whether we could get more information of these patients by isolating exosomes of these patients, because exosomes should come from the tissue. And we could indeed confirm that the LNA92A also reduced miR expression in the exosomes at 24 and 72 hours. Still, there was a trend towards significance in these few patients at 2 weeks and 4 weeks, suggesting that we indeed have an effect, possibly also in the target tissue, or at least in tissues. And finally, of course, the key question is whether we can see effects on the targeted mRNAs. We tested here 2 targets which we validated before for whole blood. This is integrin alpha-5, which is also regulated in endothelial cells. And you see here, interestingly, the time course follows a bit the miR suppression. So the miR suppression started at 24 hours, but the target only goes up later, up to 4 weeks. Here, the most significant effect was seen, and the same was true for another target, suggesting that we indeed have a target de-repression quite long-term after 1 single injection of the anti-miR. We additionally used single-cell sequencing with the idea to get further insights also on the regulated targets. And it was nice to see that predicted targets of miR-92A were really de-repressed in the LNA-treated patients. So these are LNA-92A-treated subjects compared to pre-dose, and these are placebo-treated subjects. You see here no change, and here a strong de-repression, and we could also find some interesting evidence that in some cells, particularly NK cells, also interestingly, we could find some interesting targets. ADAM10, interestingly, also Rabea, I didn't know that her data were also regulated here, suggesting that these are also targets which might be interesting and maybe also have effects on the immune functions of these peripheral blood cells. With this, I would like to conclude. So where are we currently? So the anti-miR92A, as I said, is we've finished phase 1. There's also an unpublished phase 1 study in wound healing, suggesting that there is a benefit. I would like to, in the end, show you one slide on the anti-miR132 study from Thomas Thum's group, which shows efficiency of anti-miR therapies in humans, in patients. So this is the study where they used antisense against microRNA 132 in patients with heart failure. So funny enough, Thomas was allowed to directly go into patient. To us, they told us we should go in first in humans doing a healthy control study. This is interesting if you talk to regulatory agencies, what you end up. Anyway, he shows that if you add these concentrations of this anti-miR, look, this is now a higher concentration. So ours was effective at a bit lower concentrations. We had the highest concentration was in this range. But in Thomas' studies, he used these dose ranges, and you see here also a nice inhibition over time in the plasma. This is a dose response curve. And also, interestingly, although these were really, really few patients, there were trends in NT-proBNP and some patients which improved, suggesting that there might be a beneficial effect using anti-miR therapeutics. And again, this was a single injection, which might be quite interesting in this setting to further look into the potential effects on heart failure. With this, I would like to thank the people who did the work, the entire team of our institute, and of course the funding agencies. I mentioned the DZHK support of the Translational Research Program, as well as many collaborators and people who did the work, starting with Angie Bonauer and Ariane Fischer, Andreas Seyer for all the clinical backup, and teams from industry, from Miragen, Eva van Rooij, Rabea Hinkel, and Christian already mentioned, which helped us to do the large animal studies. Thank you very much for attention.